everyone, my name is Amy and welcome to my presentation on my fourth year thesis project. My project is on evaluating sampling methods of fish mucus for proteomics analysis. My supervisor is Dr. Janina Simmons at the Aquatic Omics Lab. And just for the duration of my presentation so that everyone can see my slides, I will be turning my video off, but I hope you guys enjoy it. So here's just some information on mucus. Mucus plays a critical role in various biological and behavioral processes, and it's essential to maintaining the health of the fish. And epidermal mucus is the first line of defense against many physical, chemical, and biological pathogens that occur in the aquatic environment. And it also plays a critical role in the immune system of the fish, and ionic and osmotic regulation, reproduction, and communication. Mucus is secreted by epidermal globule cells, and it consists of a vast array of molecular components, such as glycoproteins, immunoglobulins, and pheromones. It also contains antimicrobial peptides, lysozyme, lectins, and proteases. The main components are primarily water and mucins, which are a type of glycoprotein, and the interactions between these mucins and the water is what gives mucus its viscous and slippery property. So the purpose of my study was to investigate the proficiency of protein identification of fish mucus by comparing different sampling methods for proteomics analysis. And I hypothesized that the methods using ultrafiltration, scraping, and chemical sedation combined would produce the highest number and intensity of proteins detected in the epidermal mucus. So why should we study epidermal mucus? Well, first, it is important to have the option for non-lethal sampling, especially if we wanted to study any endangered or threatened species, and this can also allow for repeated measures on individuals. It is relatively inexpensive and quick to perform, and it has been suggested that the mucus biochemistry has correlated with blood chemistry, so this can allow for a better understanding of the fish immune system. And what is interesting is that the composition and rate of secretion of mucus is influenced by many factors, both internal and external, so this could be things such as contaminants, age, sex, or stress. So this is really useful for studying changes in protein composition over time at the individual and community level, especially since mucus is continuously secreted and produced. So just an overview on the experimental design. I used a total of 50 rainbow trout, which included 10 groups of 5, and each group was separated based on 3 factors. So we had sedation method, laboratory preparation method, and mucus collection method. So we had 2 types of sedation, which was chemical and electro, 3 types of laboratory preparation, which included Bladder with just raw mucus, ultrafiltration with Amicon filters, and filtration with centrifuge Spinex tubes. And then we had mucus collection type, which included scraping, absorption, and wiping. And the reason you see that absorption and wiping is only used with filtration was due to the limitations of the other laboratory methods. So the Amicon filters were actually too small to hold the Kim wipes, so that was one limitation, but we wanted to test the methods anyways between each other. So just to go over how we collected our mucus, so the difference between scraping, absorption, and wiping. All methods we wanted to gather as much mucus as possible. Scraping just involved the use of a spatula and gently scraping the lateral sides of the fish from anterior to posterior end, just to be careful not to add scales to the sample. And then absorption and wiping were done with a Kim wipe. It was either scraped across the lateral sides of the fish using the wiping method, or it was placed on the fish for 10 seconds and allowed time to absorb the mucus. Just some pictures that we took while collecting in the laboratory. So we have scraping here, absorption, and wiping. Just a quick look at the use of filters. So both the mucus and the Amicon filters and the Spinex filters were spun in the centrifuge to separate the protein components of the mucus. So the tubes that contain the Kim wipes were added buffer, and then they were spun, and then the Amicon filters are unique in the way that they needed to be spun an additional time with the filters upside down so that the concentrate could be completely separated. So just a quick overview here of the Bladder method. So we started with raw mucus, and we had the addition of methanol, chloroform, and milky water, and we spun in the centrifuge with incubation in between until we had the 3 distinct layers: aqueous, protein, and organic phase. So our focus was on the protein disc, so we did end up discarding the aqueous and organic phases, and the protein disc was preserved for further digestion. So now we have the protein digestion step. So at this point, we'd either have the protein disc, which was obtained from the Bladder method, or the filtered mucus, which was obtained from the Amicon or the Spinex tubes. So we digested these proteins, and then they were eventually put into the HPLC vials, which were input into the LCMS, and this technology was used to determine the peptide sequences. The peptides were matched to protein sequences using Spectrum Mill and Skyline software databases, and the statistical analyses were done using Metaboloanalysts. So now on to the results. So we have the results here for my 2A ANOVA. I ran an initial 2A ANOVA that compared sedation and collection method, which had no significant results, so there is no figure included. Here on the left, you see the second ANOVA that I ran, which compared the laboratory preparation and the sedation methods. And this only had significant effects for the laboratory preparation methods. So from this, for further analysis, I ended up pooling the sedation samples just so that I had larger sample sizes, and since this was justified because there was no significant effects of sedation. So now moving on to the differences between laboratory preparation methods, I started by plotting the number of identified proteins between each method, and I performed a one-way ANOVA, which revealed no significant results. However, what is notable here is the variation between each groups, and you can see here that filtration is different than the other groups, and also note the variation here with the Bladder Method and chemical sedation. So I went on to plot a PCA scores plot, which showed the variation between each group, and you can see here that the ultrafiltration with the Amicon filtered has the lowest variability as seen with the clustered points here, and it is lower compared to both the Bladder method and the filtration method via SpinX. I then went on to graph the mean intensity for each sampling group, which had a significant p-value, and I went on to perform a post-hoc test which revealed that there was no significant differences between the sedation type, which was expected, but each method of preparation had significantly different results. So here it is quite obvious that ultrafiltration has the highest average protein intensity, and here we have Bladder and filtration, which are significantly lower. So here I have graphs for the top 8 proteins with the most significant differences between each method, and you can see that each had a similar change, and this just shows you how on an individual level that the trends were kind of consistent between each method. So here in the red we have ultrafiltration, in the green we have the Bladder, and in the blue we have filtration. So now just looking at the differences between mucus collection method, I took a similar approach and I plotted the number of identified proteins between each method, and the one-way ANOVA revealed no significant results. What you can note here is the differences between the variability, and it is obvious that lower variability is seen within the wiping group. I did another PCA plot, which showed that absorption and wiping were more similar than the scraping method, and additionally, wiping had a lower variability as seen by the clustering of points here. And then we have the graph of mean intensity, which came up with a p-value of 0.21, so it also revealed that there were no significant differences between the average protein intensity between each sampling group. And again, here you have a closer look at the top 8 proteins that have the most significant differences in abundance, and with a few exceptions, majority of the proteins you can see follow the same trend between each method. So just a summary of my results, I identified over 3,000 proteins, and over 90% of these had negative hydropathy values, which indicated that most of them were hydrophilic proteins, and the protein composition represented many biological pathways. I started to look at the functions of these proteins, but since there were no biological effects of this study, the protein identification trends were what was more important. And overall, there were no differences with sedation type, and ultrafiltration performed the highest with scraping, and it had the highest efficiency of identifying proteins with both higher intensity and lower variability. And finally, if for any reason there were limitations such as the inability to use Amicon filters, using SpinX tubes for filtration in combination with wiping seems to also be efficient. So in conclusion, the protein composition of mucus represents many biological pathways, and this is something that is really informative for immunology and toxicology, especially with studies involving pharmaceuticals such as birth control getting into our waterways. And we know that aquatic ecosystems are a vulnerable type of ecosystem, since they are at risk of exposure to contaminants such as wastewater, runoff, or solvents in their environment. And mucus is a very impactful way to study fish species non-lethally in both in the lab and the field. Some limitation to this study would be the collection methods with the tubes, of course the inability of the Amicon filter to hold the Kim wipes, and the potential for individual variation within the samples. So future studies could expand on this and test perhaps larger Amicon filters to have a closer look at the efficiency of individual collection methods, and expanding to test on different species as well. That is the end of my presentation. Thank you so much for watching. I hope you enjoyed learning about my project. I just wanted to say a special thanks to Danina Simmons and Urvi Pajankar, who really helped me a lot on this project, and I had a lot of fun doing it. Thanks, everyone.